Base excision repair, a DNA repair mechanism that helps cells to minimize the rate of mutation by several folds. Uh, Base excision repair, abbreviated as BER, is found across several different species, right from prokaryotes to humans. And interestingly, it is in prokaryotes, a much simpler system, but that forms the base to understand the base excision repair mechanism in the eukaryotes, which is a little more complex than what it is in the prokaryotes. So in this section, we are going to basically understand the base excision repair mechanism in prokaryotes. Therefore, let us look at what are the learning outcomes of this session. The base excision repair mechanism corrects base lesions that arise due to oxidation. Say, for example, the formation of oxo-G, or say, for example, alkylation or deamination. Say, for example, cytosine is deaminated to uracil. So, uracil in the DNA is an unwanted base or an abnormal base, and hence that should be corrected. Or say, for example, hydrolysis of glycosidic bonds between the base and the sugar to give rise to what is called as abasic site. So all these DNA lesions, base lesions, are addressed by the base excision repair mechanism. What we are also going to comprehend is that this entire mechanism is aided by very specific DNA glycosylases, and these glycosylases are able to identify the bases which are not normal in the DNA. The BER generally addresses DNA lesions that do not distort the DNA helix significantly. So that is something that is to be noted. BER is able to identify and correct bases by screening through innumerable normal bases using minimum energy. Where if you look at when you have 1 in 10 raised to 6 nucleotides, which is wrong, you must understand that the system has to screen 10 raised to 6 nucleotides to find out that one nucleotide which is erroneous. And this is done with great precision and with minimum energy. Therefore, this becomes one of the very good repair systems that the cells have. Now, let us look at which are the key players of the mechanism. DNA glycosylases, definitely so. And there are several DNA glycosylases which are specific for specific base lesions, followed by what is called as AP endonucleases or AP lyases, and this AP over here stands for apyrimidinic or apurinic. So when we look at the mechanism, you would understand why one should have specifically apyrimidinic, apurinic endonucleases or lyases. One of course requires the DNA polymerase and the DNA ligase to finish the repair mechanism or to complete the repair mechanism. So let us just look at a little history on base excision repair. Thomas Lindahl won or shared the Nobel Prize in 2015 for having found out the mechanistics of DNA repair, and he was instrumental in the identification of E. coli uracil DNA glycosylase, which is also called as UDG. And that was done way back in 1974 as he was looking for an enzyme that could act on uracil that is formed due to deamination of cytosine. So it is a damage. It is an abnormal base that is present in the DNA, and if this base remains in the DNA, the cell is not really affected when it is in a stage where the DNA has not replicated, but when the cell divides to form a new cell, then the template that has the U is going to be red, and therefore what is going to happen eventually in one of the daughter cells is that you would have a transition mutation, which is a point mutation, but it is something that has been responsible for and understandably, a lot of cancers has been associated with this formation of uracil on DNA thanks to deamination of cytosine. So it is a simple hydrolysis mechanism, but that has led to an abnormal uracil. So he was basically involved in a lot of cancer research and aligning cancer with DNA repair systems. And that is where he was trying to look for an enzyme that is able to act on this uracil. He was expecting it to be a nuclease, but what turned out to be was a uracil DNA glycosylase. So the understanding is this, that a citidine on hydrolysis gives rise to a uridine, and this uridine is an abnormal base in the DNA which should be removed, and it is removed by the base excision repair. So in his first paper itself, Thomas Lindahl was able to kind of outline the fundamental steps of the base excision repair. And of course, over a period of time, they were able to explain each step with its corresponding enzymes. Now let us look at the first step of base excision repair. This first step of base excision repair is carried out by the DNA glycosylases. So let us consider over here a DNA wherein the cytosine has got converted to or deaminated to a uracil. So here where you should have a GC base pairing, instead of a GC base pairing, what is present is UG. Now the presence of this UG in this DNA does not really make much of a problem, but when you have this template being read during replication, then the U will base pair with an A rather than with a G, okay, and therefore what is going to happen is there is going to be a transition from a GC to a UA. So therefore you would have a change in the base pair at this position in the new daughter DNA that is going to be formed, which is basically a mutation. So therefore to prevent this U from going into the next generation, it has to be corrected. Now the point is that this U has to be recognized by something as abnormal, or there has to be something that kind of says or suggests that here is where you have a lesion and this lesion has to be corrected. So what is observed is that the DNA glycosylases, they are observed to scan or move laterally across the minor groove of the DNA, and as it moves and through the minor grooves, or it basically associates itself in such a way that it is able to laterally move through the minor groove, it is able to, at every step that it takes, it is able to basically try to find out whether the base pair is strong. If the base pairing is not strong, then what has been observed is that the DNA glycosylase, when it reaches a base pair which is not strong, when we say not strong, it means that you do not have hydrogen bonding properly formed between the two, because the one of the nucleotides is basically an abnormal nucleotide. And so in that scenario, what can happen is that the DNA glycosylase can flip out the wrong nucleotide. So in flipping out the wrong nucleotide, as you can see, the U from here has been pushed out or flipped out, and when it flips out, the U gets into the active site of the DNA glycosylase. So therefore, let us consider that you have a situation where you can see the phosphate, the sugar over here is still connected to the uracil, but the uracil which was present over here has been flipped out by the DNA glycosylase itself. After it is flipped out, as I mentioned, the uracil gets into the active site of the DNA glycosylase, and there this bond, which is the N-glycosidic linkage, that is, the sugar forms an N-glycosidic linkage with the nitrogen base. It is this N-glycosidic linkage that is broken by the DNA glycosylase. So that is the catalytic activity that the DNA glycosylase carries out. And therefore, the end result is that After the glycosylase does carry out its catalytic activity, one observes that at this point you would have the uracil removed, and because the uracil is removed here, there is no base. And so this site is called as an a-basic site, but more effectively it is called as a pyrimidinic site if a pyrimidine is removed, or a purinic site if a purine is being removed. So basically what is created over here is what is called as the AP site. Now, when you have no nucleotide at this point, again this is an indication that there is abnormality in the DNA. So this is another way of signaling to the repair system that there is something wrong over here, and hence this has to be taken care of. So effectively what we are understanding is that the DNA glycosylases screen the DNA for the abnormal nucleotides. Now these nucleotides could have been formed because of oxidation, or because of alkylation, or because of deamination. It could be anything, but they are able to screen out. So there are specific DNA glycosylases present for specific type of lesions. So it has been observed that there are about 8 to 11 different DNA glycosylase, and each DNA glycosylase belongs to a certain family depending on its structural characteristics. But the understanding is that DNA glycosylases are able to identify an abnormal base and remove the abnormal base by cleaving the N-glycosidic linkage. So the backbone of the DNA is not affected by the DNA glycosylase, but the base which is wrong is excised out. Another important fact that has been observed is that some DNA glycosylases are monofunctional. Now, what does it mean? It means that it only has the catalytic activity of breaking this glycosidic linkage. What is also found is that some DNA glycosylases are difunctional, which means that apart from the glycosylase activity, they have an added catalytic activity of a lyase. So let us understand what that the second activity of the enzyme enables. So let's say, for example, this is one strand of the DNA. So what is observed over here is a sugar. This will be over here a phosphate and another phosphate over here. So this is the two diester bonds present. Now, what the glycosylase has already done is that the damaged base has been removed. So after the damaged base is removed, okay, what remains is the sugar. As you can see, what remains is the sugar. So the base has been removed, and what remains is the sugar. Now the enzyme in its active site has an aspartate, which is active, and a lysine. So the aspartate and the lysine together, through a series of reactions, are able to carry out a beta elimination. And by the beta elimination, two things will happen. One, the lyase will break the bond that is there over here between the sugar and the DNA. So what you will get as one of the products is this DNA with an OH group. So the 3' OH has been formed over here, and the other part of the DNA strand, remember this is a single strand. So it is not completely dissociated. It is still attached to the other DNA strand over here. But what is definitely the case is that the sugar is still in this form, a broken form, be attached to this part of the polynucleotide strand, while this part of the polynucleotide strand is free from the sugar. So the lyase activity has enabled break one of the ester bonds that is present between the sugar and the phosphate in this strand, in the parent strand. So understandingly, what we would observe is that because of the presence of lyase, the lyase is able to say cleave this bond, okay? And because it is able to cleave the bond, what happens is a 3' OH group is formed over here and a 5' phosphate. So there is a nick that has been created because of the breaking of the bond. So you would find, so sorry, it is not this bond, it is basically this bond. So this is what is going to happen. Now, it is not necessary, however, that all the systems have the DNA glycosylase acting as a lyase, but it has been observed to do so. So the understanding is that, I'm repeating what is said, is that it enables forming a bond with the sugar. That is, the enzyme forms the bond with the sugar, leaving a DNA with a 3' OH. This is what is the case. However, now, having understood what the DNA glycosylase is practically doing, let us go to the next step of the base excision repair. So we come back to the situation where after the DNA glycosylase has removed the abnormal base, what remains is the sugar phosphate backbone of that strand which had the abnormal base intact, but the base is removed. Now, such sites where it is apurinic or apyrimidinic is recognized by another enzyme, which is the next enzyme in the BER mechanism. That enzyme is called as AP endonuclease. Now, this AP endonuclease, as the word suggests, okay, is a nuclease. That means it can cut a phosphodiester bond anywhere within, but not anywhere within. It is specifically at apurinic or apyrimidinic sites. So it can identify apyrimidinic or apurinic sites, and once it identifies such sites, it cleaves the 5' end of the phosphate sugar moiety of that nucleotide which has been removed. Correct? That means at the 5' side of the AP site. Basically, that is the thing. So you would eventually have this bond being removed by the AP endonuclease. And because this is removed, what is observed is that here you will have a 3' OH and this will be a 5' phosphate because a nick has happened because of the AP endonuclease. What happens next is, therefore, you have now a flap being formed because you have this phosphate being removed from the sugar because the phosphodiester bond has been broken. What is observed is that this in turn can be removed by a exonuclease. So if you have a 5' 3' exonuclease, it is going to cut the sugar at this point, okay? And when it cuts it at this point, what you observe is that this phosphate and sugar that was part of the original nucleotide has been completely removed. So now what is seen is that there is a gap in the phosphate sugar backbone as well, where there was an abnormal base. So eventually what is happening is that not just the base is removed, but also the phosphate sugar that was associated with the base has been removed. So by the second step, therefore, what is understood is that the sugar phosphate of the base lesion is also removed and it leaves a gap. Now this gap is something that has to be filled. So we go to the next step of the base excision repair mechanism, which is carried out by the DNA polymerase and ligase. So coming back to the same situation where there is a gap and you have a 3' OH and the 5' phosphate available, the 3' OH is bound to by the DNA polymerase. So you know that DNA polymerases always will use a 3' OH as a nucleophile to introduce a new nucleotide. So this DNA polymerase brings in the correct nucleotide, correct? It forms a phosphodiester bond over here. It brings in cytosine, which is going to form the complementary base pair with G. So where you had a U earlier, now you have a C, which means that the DNA polymerase is filling up the gap, okay? How? But filling up the gap by introducing the right nucleotide or the right base. And so this is done. However, it can only add the nucleotide, but it cannot seal the final, it cannot form an ester bond between two existing nucleotides. So therefore, a DNA ligase will come and seal this to form this. Now, therefore, what you have is a DNA which is back to normal. So a uracil which was present over here has been removed and now you have a correct normal DNA that is formed. So this is what the mechanism of base excision repair is. Since it was just one nucleotide or maybe at the maximum two nucleotides which could have been removed by DNA polymerase 1, if it is DNA polymerase 1 that is binding, it can also keep removing nucleotides from here for a certain distance and keep adding new nucleotides. If it is a short patch, then it is what is considered to be a short patch BER, or base excision repair mechanism. But you can also have another system which is called as long patch base excision repair mechanism. Now let us consider that you have the DNA, and this over here is the place where there is a base lesion. So this nucleotide is a wrong nucleotide or an abnormal nucleotide and has to be removed. So the DNA glycosylase comes, removes the nucleotide, and you have the AP endonuclease coming, creating a nick, and you have the DNA pol coming, and now it is synthesizing the new DNA. But what is observed is DNA pol 1 will keep removing certain nucleotides. So a longer patch is being removed and synthesized by the DNA polymerase 1, while this that is forming is a flap. So you will have another nuclease which will come and cleave this DNA, because a new DNA has already been synthesized. After it completes that, then you have the DNA ligase coming and sealing it. So this kind of base excision repair also is observed more in the eukaryotes than in the prokaryotes, but it is definitely observed. And so this is what is called as a long patch BER. So let us conclude. Understanding the BER mechanism has shown that there are specific DNA glycosylases that can act on one or more specific DNA base lesions which are present due to oxidation, alkylation, etc. The DNA glycosylases scan to identify the specific base lesions by flipping the base to remove it from the DNA. The AP endonuclease and the exonucleases remove the phosphate sugar of the damaged lesion, leaving a gap, and that gap is filled by the DNA polymerase, and the final seal is done by the DNA ligase to complete the process. Thus, the base excision repair mechanism is another very good recipe for survival of the cells, targeting only the base lesions and removing just the base lesion or a few flanking bases to replace with normal bases in the DNA. Thank you.